And I'm delighted to introduce our next speaker, Elizabeth Purdom. She is Associate Professor in the Department of Statistics here at Berkeley and also the Faculty Director of the Center for Computational Biology. And she'll be talking to us about multimodal single-cell sequencing data for integrative analysis. So thanks very much, Elizabeth.
Great, thank you. I'm gonna dispense with my mask for the talk. Yeah, so I want to thank the organizers for organizing the conference and inviting me to talk. And as you can see by the large number of talks we've had about single cell sequencing data, it's clearly creating a large number of new opportunities in exploring problems in biology, evaluation of cell type diversity, developmental changes, cancer evolution, being able to go from sequencing a mass of cells to sequencing an individual cell is clearly very transformative for certain questions. I was going to take a moment to think about this a little bit more from the point of view of a methodologist. When I got my PhD, I was working on microarrays, which were kind of big data at the time, measured huge number of features for the time. And, but at the same time, the number of samples was modest. And of course, since that time period to now, we had these vast data sets in other areas where they just sort of scoop up data as we go about our social interactions and on the internet and so forth. But for me, at least, most of my projects that I'm involved with, the size of the data set is stayed sort of stubbornly modest in terms of the number of sample sizes. So it's sort of a different way of thinking or analyzing the data than some of modern advancements in thinking about, how to deal with Google social networks and so forth. So one thing that's interesting to me is that in working like single cell data, at least for me personally, is a slight paradigm shift in the sense that now we easily have hundreds of thousands of cells. You saw from previous talks, you can easily have 1 million cells. So for me, this has been kind of exciting in terms of being able to think differently about my problems from when I had only 100 samples. So in thinking about what to talk about today, I decided to talk about a project that I worked on with a student. This sort of exemplifies for me sort of an example where I kind of shifted paradigms and ways I would have thought about the problem. So that's just sort of my big picture overview of my motivation for talking about this. What I'm going to talk about specifically, I want to give a little bit of a background to. I think a number of the talks today have been focused largely on measuring mRNA, the transcriptome. But there are single cell technologies for, if not all, many of the bulk technologies that existed before for sequencing. So for example, there's many areas in epigenetics where we can do single cell sequencing and expand beyond bulk as well. So examples are things like open chromatin, methylation, proteins binding to DNA, and so forth. And I'll talk specifically just to give a little bit of background about open chromatin to make sure we're all familiar, because this is one that's actually become really fairly frequent in single cell sequencing on ATAC-seq. And the idea here is, of course, our DNA is quite large and is generally to be able to fit compactly in the cell is tightly packed and closed. But in order to be able to transcribe genes, that packing needs to open up so that the gene can be accessed by the transcriptional machinery. And so open chromatin basically is going to be trying to evaluate where is the chromatin open versus tightly packed. And so you get reads, and I won't go through the technical procedures, but you get reads more from the DNA that is accessible. And so you might have something that looks like this, where in the tightly packed regions, you don't have a lot of reads. And in the open regions, you have these peaks. Okay? So that's just one example, and it's an epigenetic trait. It's not something built into the DNA, but something we want to measure. And it clearly has large interactions with transcriptome because, of course, regions that are going to be transcribed will have areas open, but other more distal regions can also be open for enhancers and things like this that are necessary for transcription. And so many of these are often done they're often not completely in isolation, but also in conjunction with doing transcriptome sequencing from the same biological system. So an area of great deal of interest is how to combine these different modalities. And there's kind of different settings for which this can happen. One setting is to do the two different technologies, the mRNA sequencing and the ATAC sequencing, for example, on two different experiments from the same basic system. In other words, on different cells. You burst open the cell and so forth, so it's easier to do it on separate cells. And there's various techniques for trying to analyze and combine data when they're, so to try to find commonalities. This usually requires you, even though these modalities are not necessarily measured on genes, we have these peaks, which may or may not be over gene regions. These modalities get summarized into something that has to do with genes, and they're related up by looking at the gene measurements from the RNA and the gene measurements from the peaks, for example, and putting those together for shared patterns helps you link up these two data sets. So that's kind of option one in terms of the type of experiment one might have, but as was very briefly mentioned in the previous talk, there are also technologies that sequence the same cell in multiple ways. So what do I mean by that? I'll take the example, the running example here of ATAC-seq with mRNA. And there's several technologies that actually from the same cells try to measure both of these quantities. So we'll have cells, and on each cell, we can have measurements of the gene expression, but we can also have those peaks, the counts of the peaks. And so one question is how to jointly analyze these two data sets. As this sort of toy cartoon might sort of illustrate, the peaks are generally can be of a sort of different nature than the genes, more sparse and so forth, and trying to put them together in a way that's not just combining them into one big matrix to try to get a unified representation. Another goal we were particularly interested in is thinking more holistically about the entire experiment. One might run joint on a certain number of cells, get both modalities, but it's also much easier and a more tried and true system to run them separately. So we might on the same system also have cells that only have the gene expression data and cells that have only, for example, the ATAC, the open chromatin measurements. And so we're interested about how to integrate this data, this kind of data when it's collected on multiple platforms. And of course, you could have multiple versions of each of these going forward. And sort of as a side goal, we also are interested in not having to summarize the peaks into genes, but actually use the full peaks, right? Because especially since you can have distal elements that are important for expression, we would like to be able to not have to throw out that information. So not a primary goal, but it was something we were interested in. So what do I mean by a unified representation? So I'll kind of walk you through the ideas that went into the particular strategy we did, but I would like to say this is a very active area, and these kind of ideas are in a lot of different methods, both that were coming out at the same time as ours and in other related areas. So the basic strategy, what we mean by a unified representation is that we think that there's some underlying latent variables that are a property of the cell. We can think of them, if we like, idealistically embedding the true biological processes, and that these to be able to get to mRNA, and then what we of course measure is a process that these latent variables go through to create the count data that we see from the RNA. And similarly for open chromatin, that creates another process that gives us the data. And so for these same yellow ones where we get to observe both the end products of both, but the idea is that they come from one shared latent variable. And if we did on these cells get both, it would be the same. In other words, there's these latent variables that we want to use. And what we'd like to do then is estimate from the data these Z's. So we start with this set of data and we want to get some estimate of these latent variables for all the cells, regardless of which platforms or combination of platforms we have measured them with. Once we have those, then those can be used for downstream tasks for things like visualization and also, of course, clustering to try to identify subtypes. Okay, so I'll start first with the where we started and then kind of build up from there. So the first idea was built on some ideas that had become current in single cell. And again, this takes advantage of the large number of cells. So this is kind of going back to my original point. I think embarrassingly, that's actually my phone. So unfortunately, even though it's silenced, it decided to go off anyways. Okay, sorry about that. So using ideas of variational autoencoders, for example, SCVI, which is was created by Nir Yosef's group and others here at Berkeley, using variational autoencoders to try to take, for example, just with one data set to try to estimate these latent variables. And they rely on using neural networks to approximate the posterior distribution of these latent variables. And there's some generative model, which is the portion over here of how you go from the latent variables to the counts, and that you can encode for various different models, such as have been talked about with previous speakers, such as zero inflated negative binomial. There's also multimodal variational autoencoders, VAEs, which have the idea of taking different modalities, such as we had before, and actually developing different neural nets for each one to encode how you would get the prior distribution, and similarly have a generative model like I described before. So that was our first idea, was to basically do a straightforward kind of implementation of these strategies. And one problem we found, at least in the data that we were looking at, was in fact the open chromatin data was really sensitive to how the generative model we chose to use in a way that the RNA sequencing data was not. And so in particular, this is just one example that's easy to draw, but this was true with other types of approaches. This is just sort of a standard analysis in Seurat using their standard tools for chromatin, ATAC-seq, of the kind of, and a visualization of their reduced dimensionality, while another strategy that used a very different approach, cystopic, got something that was much more well-defined in terms of its subtypes. And so these kind of problems made it very difficult, made this strategy actually not work well for us. And so we adapted further to try to make advantage of what Cystoptic uses, which is a latent Dirichlet allocation model. So if you're not familiar with that, I'll just briefly describe it. It's created in the context of documents, where the counts you observe are the number of frequency of words in a document. And you assume, and documents are expected to have various different topics that they're encoded in. So you assume that the counts you observe are actually a mixture, in other words, an additive sum of the counts, the words being used for different topics. Okay, so there's some parameter, this theta, which gives you the proportion for any particular cell or document of each of the topics that are done. And for the biology setting, you can think of this as different biological processes. You know, some RNA is going for one process, some is going for another process. You add it up is what you see. And so theta, that's idealistic, obviously, but theta C would be the proportion of those processes that this particular cell needed. And then you have a parameter that converts, that lets you know, given each topic, how much do you get of each gene. And so by putting those two together, this is independent of cell. This is just shared across, say, all of mRNA, and this is per cell. By putting those together, you can actually get your observed counts, which is going to be, say, a multinomial across this combination. So this is the cis-topic strategy. In order to make it work in our multimodal type of context, we use sort of a VAE version of LDA, where we take a transformation of our, we assume that our parameter that's of interest, that's the proportions, that's kind of our latent variable. We assume that it's actually a softmax transformation of those Z's we were actually interested in. So we're still back to our Z model. And when we have those Z's, we can transform them to get them into proportions. And this is using a standard softmax transformation. And then instead of using a Dirichlet prior, we're going to be assuming that we're going to be approximating the distribution. Instead of thinking about the prior of the thetas, we're going to be trying to model the prior of the Z's. And then our VAE will then be modeling or approximating, because it's variational autoencoder, approximation the prior distribution of the Z's with a multivariate normal. So putting those pieces together is the model that we developed for the multimodality, where this is the same picture as before, only now instead of using the previous generative model, we're going to put in the parameters for the, we're going to be instead modeling the LDA there. And so just to give you a sense of what this looks like in practice, how do you actually use this? In the end, what we get out are estimates Z for our different modalities. And so, for example, this is a data set on the mouse Cortex, where there were separate, single cell mRNA and ATAC-seq, and then also snare-seq on another, from another group, but also from the same system. And so we look at integrating these together. Here is just a representation, UMAP representation of those Z matrix, those Z latent variables. They're in a much higher dimensional space, but this is just a two-dimensional representation. And so on the left here, we can see them color-coded by the different data sets they come from. And here we can see them color-coded by the clustering, the known, not known, but the cell types that were identified from the paper. And in the case here, we identified them de novo from our representation. And so we can see that they well categorized. And this is true in other data sets. We also, so we looked at various different things. One thing that's sort of nice about working with these paired data is we actually know, of course, that they come together, but we can pretend we don't know. And that gives us sort of fake data. It's real data, but we actually know which ones should go together. We can pretend we don't, and we can analyze sort of how well we do at being able to put these data sets together. And so, and why do we do this? I mean, there's a lot of different reasons. One is these are very large scale groups that are pretty hard to not get fundamentally. So we wanted to look at a more local level, how well they were doing. And so we're looking sort of, if we use these techniques, in other words, if we pretend, going back to, oh, it's way back here. Okay, if we pretend that our red and blue data sets, and other isolated data sets, they're paired data, we'll pretend they're not, and we'll put them into our model and see how closely we're able to align them together. And so this is just basically how well we're able to align them with a higher proportion, meaning that you get more of the ones that should be together, close together. And this is our strategy here, and we've compared it to some other strategies. As I said, this is a busy and growing area. And so these are two different data sets where we've done similar things. And we can see at the very more detailed level in terms of which cells are actually close to each other in our latent space. Are we doing a good job of actually pairing them together and saying which cells should be similar to each other, that we're doing better than these other strategies. And going back to my side note, if I add some more lines in here, we also tried looking at different ways to actually pre-process the data. And indeed, this line here is, that I point with an arrow, is if we instead summarize them based on genes with our model, with Cobolt, and we ignore some of that peak. And satisfyingly, we do get higher, we do better when we actually use the full set of information and not just the gene strategies. So, so that's kind of an overview of how our method works, but also just sort of these ideas, like how people are approaching these multi-modality data sets. And so I guess maybe just to close, I'm going to go back to my original slide saying, oh, I have all of this data and how nice this is. And maybe comment on that a little bit more in the sense of many large census or atlas projects, the goal is to really discover sort of the diversity of cell types and to get as many cells as one can. And in that setting, for example, as part of a mouse brain atlas project that I was tangentially sort of related to, to get enough cells, you might have multiple mice that you use as donors for the cells that provide gene matrices. And they're going to be hopefully homogeneous, genetically identical, very similar. And what's usually done is, of course, we're going to group them all together. We might try to look for batch effects. We might try to remove them. We'll try to be sophisticated about it. But in the end, they're combined together in basically one very large matrix, which is more or less what I've done here. However, when we move beyond and we try to think more about human health setting, and this is maybe a little bit more like the setting that was in the previous talk, it's a kind of different setting. In this setting, we would have multiple patients, and each one would provide single cell RNA sequencing data. And we no longer want to have homogeneous, or we no longer will have homogeneous groups, despite my poor little cartoons. And they should be quite different and have population variability. And so we want to be able to capture that variability, not remove it, and be able to determine what of that variability is due, for example, between different phenotypes of interest, disease versus control, and what of that is population variability and so forth. And so in some sense, that brings us back full circle to my modest data set sizes again, in the sense that the number of data sets, these data sets are growing as more and more of these types of experiments are run, but we're still talking about, modest cohorts, 40, 100 patients and so forth. And so one area that we're quite interested in my group is actually this question of what is the right way to combine these data sets together, which is fundamentally a question that's going to vary for all the different kinds of single cell type of questions we might want to ask of the data. So there's my asterisk to my excitement previously about the data set sizes. And I will finish by saying this is the work I present today is the work of my student, Boying Gong, but I wanted to recognize that I have very nice collaborations in general on lots of different single cell sequencing experiments, a number of people in this room. And so they've also been helpful. And this is the paper of this particular method that came out in Genome Biology last year. Okay, I think I'll stop there. So, any questions?
Okay. So thank you very much. So you are looking at the latent structure of the different omics modalities and single cells and try to somehow find which is the common latent structure. And somehow I can imagine that there is a strong relationship between chromatin accessibility, a gene expression, so that this latent structure exists. But I can also imagine that very interesting question is which are cell types of subsets of cells in which this is not happening. So there is something that is not, I mean, there's a structure that is not shared, right? Because that's, I don't know, indicates some kind of a regulatory problem maybe. So is your methodology is also able to identify this kind of different type of latent spaces across modalities?
Yes, so the yes and maybe no. As your question continued onward, I had to rethink it. But fundamentally, this model here has this B, which goes basically from your latent variable into what you get for each gene. And I didn't explicitly say that, but when we move to a multimodality model, this B is going to be different for every modality. So you can have consistent strategies for how, how your latent variables translate into the modality data. And that's modality specific, right? So you could have, despite using process 1, it could be that process 1 doesn't turn anything on in the chromatin and it does in the RNA, for example. Now you started, as the question moved on, to talk about cell type specificity. And I think the cell type specificity would instead have to be encapsulated in these proportions, right? And those are per cell. So while we're not modeling cell type per se, you could think that there's a hierarchy of these latent variables that amongst different cell types, they will have different underlying latent variables. And in some sense, that goes, I guess, back to the prior, which you're trying to estimate empirically.
Okay.
So this is maybe a little related to Anna's question, and I think I'm constructing my answer in my head, but I'll ask the question if you want to add something else. So is there any intermediate inference objects in your model that helps you learn interactions between gene and peaks so that you can learn regulatory programs?
Yes, so this is something that we've explored, but not really gotten to prime time. So which is this question, this sort of comes into this B matrix where you get a different one for each modality. And that's kind of work in progress as to how successful that is, honestly. So I can't say more than that, but that's our strategy and something we're investigating right now is how you could use those to try to have, to make explicit those relationships. Yeah.
Yeah, I mean, following up on the same sort of theme, obviously other methods make strong assumptions, right, of sharing features and, promoter accessibility, gene expression, and you make another assumption, and I assume you have pair data to basically learn those relationships in some way. I was wondering whether they're useful intermediates. I mean, one of them is, local proximity. Even if you don't make promoter accessibility, things that are near a gene might be more related to that, or other information, whether there are ways to sort of encode this prior knowledge column quote in your model, because I think it's unconstrained in a way, right? You could have shared latent variable of things that are even on different chromosomes. And there's wondering whether that's something useful to explore or possible to encode in this framework.
That's a good point. I did come in with the bias of struggling with trying to work with ATAC-seq and defining it in terms of genes. In this, working with a collaborator and what is the distance? How far does it have to be away? So on and so forth. And so I did come in with the bias of oh, I don't want to do that, right? I really want to just deal with the peaks and, figure out what they tell. But I agree that in some ways the model is overly open in that respect. So again, I think this would come back to putting constraints into these B coefficients, and but it's true. I thought about more like Sunda's question, how do you interpret them when you get them out? And so I hadn't really thought about that from a regularization point of view, but it's a good idea.